Okay guys, so we are going to continue with the two main type of adaptive immunity, which is known as humoral and cell-mediated or cellular immune response. We're going to start by looking at the humoral immune response, which is created by a category of lymphocytes known as B cells. Now, B cells are going to be activated when they meet their corresponding antigen. And this is what we described previously. Right before they meet their corresponding antigen, they're identified as immunocompetent, but they lack their activation. So they're considered to be naive. So as soon as they do interact with their antigen, then they become activated and form a category of cells referred to as the effector cells. And then effector cells also form a category of and form memory cells. Now, effector cells in the case of your humoral immunity are called plasma cells. So effector cells that we use, we learned about it earlier in the previous PowerPoint. Effector cells are a generic term we use as the cells of the adaptive immunity that create a very significant immune response. But in a case of humoral immunity, those cells are referred to as the plasma cells, and then memory cells are going to be the same as what we discussed before. So, what are the jobs of the plasma cells? So, plasma cells, one of the key concepts that you guys need to be very clear about is that there are the only cells that are going to be producing antibodies. No other cell of your immune system will be producing antibodies except your B cells, which are going to carry out an immune response against the infection. The antibodies recognize that they are specific to every antigen, hence they need to be activated, go through clonal selection. And what they do is they mark any antigen or pathogen for destruction by other components of the immune system. So please do understand that antibodies themselves do not destroy your antigens or do not destroy your pathogen. They typically mark them to be destroyed by the rest of your immunity, which is either adaptive or innate immunity mechanism. Again, inflammatory response, your other category of white blood cells such as dendritic cells, macrophages, neutrophils are the ones that are going to be basically destroying your antibody marked pathogens. Memory cells I already discussed with you as well. If you look at them, some of the effector cells become memory cells that are going to act as the guide to look for any type of antigen that was already recognized. And if that recognition takes place, then they create a much stronger immune response against the same pathogen. Here's a picture. Hopefully this helps you clarify the steps involved. So what we have here is we have three different types of B cells that are immunocompetent, but they're considered to be naive. So in this case, among the three different type of B cells that we have, the central one is the one that recognizes the antigen. So as soon as this recognition happens, then you have a naive cell becoming activated. When they are becoming active, then they start to do what? Proliferate, which means they're going to form more and more of their own clone. Now, there's two main categories of these effector cells that are going to be created through the proliferation process. One type are your plasma cells, which you see right here. They're going to produce lots of antibodies, and these antibodies are going to do what? Basically target, if you want to again think about them, target pathogens for what process? destruction. On the other hand, you also get another second category, or the second category of cells called the memory B cells, which you see continue to go down, and these are the cells that are basically remain in the system. They circulate in your body until they interact with the same pathogen as the original one you saw here. So this is the second exposure to the antigen. Notice that you produce that memory cell that you have here, which is right up here as well, is going to produce lots and lots of copies of the plasma cell again, which produces lots and lots of antibodies, and that is going to basically create an immune response against the same original antigen that you had. Do recognize that there is always some memory cells left behind for a possible exposure to the same pathogen later on, which will trigger the same immune response again. Now, working on this whole concept of first exposure versus second exposure, we have something called primary immune response and secondary immune response. Primary immune response is a condition where you are, again, keyword here, you are doing the first exposure to the antigen. First exposure to the antigen. So there is a lag period of between 3 to 6 days because these cells need to go from naive cells through the clonal selection and then proliferation, which takes about, again, 3 to 6 days to happen. So when you are going to reach the maximum amount of antibody or the peak level of antibodies, it takes about 10 days. Antibody levels after that start to decline after they deal with the infection. So, two problems. Well, not necessarily problems, but characteristics of the primary immune response. It has a lag time, and it takes a long time for it to be activated, and the level of antibodies are relatively low. On the other hand, let's say you activated the immune response, and part of that immune response was to create your memory cells. So memory cells that you created are going to stay inside the body, and then when you have re-exposure to the same antigen, then what do? You use your memory cells this time. Therefore, you're basically creating something called an immunological memory. The response is within hours rather than days. Remember, 3 to 6 days lag days. You reach your peak antibody in about 10 days, but here you reach in about 2 to 3 days, and the levels are going to be much higher. And antibody levels are going to remain high for a longer period of time. So in this picture, you can kind of see it. In this picture, the blue line represent primary and secondary immune responses. So you produce antibody, but notice the level is relatively low, right? It's about here. Again, it declines going down. You have the second time being exposed to the same antigen, and notice the amount of antibody you're creating, somewhere up here. So lots of more antibody, and look at the time. This is the time, and again, very much faster response versus a good distance, which is about 10 days to produce a recognizable amount of antibody. So these are extremely important because this is the way we protect ourselves against possible viruses and bacteria that can cause major problems with our body because we won't have enough time to create the primary immune response. So when we're talking about vaccination, what we're doing is we are forcing your body, let me explain this. You are forcing your body by giving the vaccination here. You're forcing your body to do what? To create your primary immune response. So vaccination is not the full full virus or a bacteria. It's typically a fragment of the virus or bacteria, or it is a dead bacteria, which basically means it has no ability to harm you, but it can, because the antigen is present, it can activate your primary immune response because the antigens are there to be recognized. So when you get your vaccination, you create this, right? But what you're doing is you're creating your memory cells. So sometime later in your life, if you get the exposure to the actual virus or bacteria, then what does your body do? In a matter of hours, it creates a significant response, and as a result, it doesn't really give the chance to the infective agent to root and cause damage in your body. This process that I'm describing as a vaccination is an artificially acquired immunity, versus you have naturally acquired, which is where basically you get exposed to a bacteria or virus, and through a second exposure, you can defend yourself. But there was no vaccination for it. Now, a quick note about the antibodies. There are a total of 5 different types of antibodies in your body. The only thing you need to know is the name of them: IgG, M, A, D, and E, 5 classes of them. They do contain something called a heavy chain versus a light chain. So right here are your light chains in light blue, and the darker blue color are considered to be the heavy chain. The tips of the light chains are identified to be where your antigen is going to be binding, and it's going to be different from one antibody to another. But what is identical in all of them is majority of the light and the heavy chain, light and heavy chain of your antibody. So you can see the similarities. They are very similar, even though they might be bigger or smaller. There's very similar structures or components within these 5 categories of antibodies. Here is an overview of humoral immunity. What we are showing here is you have the antigen on this side, the recognition by the antibody. You could either, when they interact, you can neutralize anything that could possibly cause harm, such as toxins or actual viruses, which is what they're representing here. You can have agglutination, as we described for your red blood cells. You can see right here. You can have precipitation, where you actually literally pull out the antigens from the system by precipitating them out, solidify them in another sense, pulling them out of the fluid. We have complement system, which basically you have the antigen right here on the surface of your bacteria, or let's say it's bacteria, but typically it is going to be bacteria, bringing the complement system, putting out holes and pores, water coming into your cell and causing the bacteria to burst. In these 2 scenarios, or even, I should continue. This is where your bacteria is burst. In a case of agglutination, neutralization, and precipitation, all of them can basically mark your antigen by the antibody and mark them for the process of destruction. As you can see here, you have phagocytosis, which basically you eat up the bacteria, or you have an inflammatory response, which carries the same process and destroys your antigen. Moving on to the second category of immunity or adaptive immunity, we have cellular immune response or cell-mediated. Again, the key word is when we talk about cellular immunity, we're talking about T cells. One of the main goals they have is they attack cells of your own body that has been infected by either viruses or bacteria, or even cells that are considered to be cancerous or abnormal. In these cases, these cells, these T cells, what they do is they force the infected or damaged cell to suicide or to self-destruct or suicide through a process called apoptosis, or by basically creating a bunch of holes inside the infected cell. Some of the other cells are responsible for maintaining the response created by your body. These are referred to as T helper cells. They control the amount of chemicals such as cytokines that are being released in the body. And through that, they control how adverse the immune response is going to be, or how effective your immune response is going to be. So let's take a closer look at these. When we're discussing cellular immunity, or as I mentioned, cell-mediated immunity, they are relying on T cells. There are two different categories, two main different categories. CD4, which is also known as T helper cells, and CD8, which is known as cytotoxic cells. Both of these are going to also form memory cells. A branch of it can form memory cells, which carry the same identical function as B memory cells, which basically flowing around and making sure that they are guarding against the same pathogen for later in life. Picture looks very confusing. Don't really need to know all the detail. Couple things that you do need to focus on is here. So we talked about antigen presenting cells. If I want to activate my T cells, so notice the word activation, you need to present your antigen, which is right here in red, and right here in red. You need to present them to your T cells, the two categories of T cells, using the APC categories of cell. You don't need to know the histocompatibility complex. That's a little bit more detail. When antigen are presented, as you can see here and here, then the naive cells become active, and then it starts to proliferate, which basically means make copies of itself. And that copies form your effector T cells. So, looking at the process of T cell activation and proliferation, activation, as I mentioned, relies heavily on antigen-presenting cells, which was their dendritic cells. That's one of the main categories. Oops. The other main categories were your macrophages. This is kind of a review from the earlier lectures. And even B cells can act as antigen-presenting cells. When they when these APC present their antigen to your T cells, it causes a signaling pathway that eventually leads to activation of T cells. At this stage, your T cells can basically get activated, undergo proliferation, and create a response. Again, the categories of T cells that are being produced, I'm going to go to the next stage for just a second. There's two main categories. You can say three different type of categories. One categories are known as T helper cells, and I'm going to just list it as number 2 here. Number 2 was your cytotoxic cell. And if you want to go a little bit more detail, both of those can eventually become memory cells for future recognition. Now, what is the function of T helper cell? What is it so important? It activates B cells and other categories of T cells. It helps both of the T and B cells to proliferate. And it secretes something called cytokines, which are going to recruit other immune cells. So look at the vocabulary, guys. T helper cells, which means they are going to help the process along. They don't necessarily do the work, they help. They assist the process to go along. Cytotoxic cells, which is the second main category of your immune cells or your cell-mediated immunity, are considered to be extremely important. They're the ones that are basically going to force the cell to destroy itself. These cells, for them to be the target of your cytotoxic cells or T cells, TC cells, they have to be virus infected. So one of the characteristics. They should be cells that have intracellular bacteria or parasites. Basically, within them, they have bacteria or parasite residing in them or damaging them. Cells that are identified as cancerous, and any foreign cells that are being basically introduced through the process of transfusion or transplant. So a lot of time when people are basically undergoing transplants, they are taking a lot of medication to suppress their immune response. How does your cytotoxic actually affect and destroy these cells? As I mentioned earlier, they create pores inside the cell, and these pores are basically a signal for the cell to destroy themselves or cause cell death through a process called apoptosis. Here's a picture. I really like this picture. This is an actual scanning electron micrograph of a cancer cell being targeted by your cytotoxic cell. So here's your cytotoxic. Here's your cancer cell. Right here, this is another picture. You have your cytotoxic cells here and a target cell here. I believe this is an infected cell. And what they're doing, again, they're creating pores inside the membrane. And when these pores are done, what happens is the fluid starts to come in, and that fluid entry causes the cell to basically increase in size and burst. Last couple of slides are focusing on two of the main medical conditions associated with immunity. First one is created by a virus called Human Immunodeficiency Virus, or HIV. It creates an immune disease called Acquired Immunodeficiency Syndrome, which is basically the body is unable to defend itself. It's characterized by severe weight loss, swollen lymph nodes, significant amount of sweat at night. It is transferred, HIV is transferred to any type of bodily fluid, and also through semen and vaginal secretion. When HIV enters the body, one of its targets is T helper cells that are basically monitor and control your immunity. And through that, they suppress the immune response. This suppression of the immune response basically leaves you open to other type of infection identified as opportunistic infections, such as pneumonia. I always mention this, that patients suffering from AIDS do not usually die from AIDS. They die from secondary infections such as pneumonia, which is relatively sad. Which is sad, not relatively sad. It is sad. But do remember that HIV is such a horrible virus because it's attacking the source or the means for you to defend yourself. So it doesn't affect any other parts of the body. It affects the part that is supposed to be defending your body. Lastly, we had autoimmune disease. Autoimmune disease are basically your body loses the ability to distinguish between your own cells versus cells that are considered to be foreign. So in many cases, what it does, it starts to identify your own cells as foreign and mark them for destruction. Some of the diseases that are considered to be autoimmune disease are rheumatoid arthritis, which is target the cells between your joints. Multiple sclerosis or MS, which destroys the white matter that are formed around your neurons. Graves' disease, which causes the disease called hyperthyroidism, and we'll talk about that in Bio 251. And type 1 diabetes mellitus, which is where your immune cells attack and destroy a category of cells called beta cells within your pancreas, responsible for production of insulin. As far as material goes, this is for your lymphatic system. And that's it. Thank you.